So the next unit, that is unit 3.4. Here we are going to discuss about the structure and the function of endoplasmic reticulum and its role in the protein segregation. It is a part of your unit paper 1 unit 3 and the subtopic unit 3.4. The endoplasmic reticulum is comprises of the complex network of the membrane, as you can see in the diagram, which is basically covering the much of the part of the cytoplasm. Okay. As per the endosymbiotic theory, which we all know, the endoplasmic reticulums are thought to be evolved from the invaginations of the plasma membrane. Now, as you can see in the diagram, the space which is enclosed by the endoplasmic membrane, the one which have been shown in the brown color, okay, the internal space which is enclosed by this membrane is called as lumen, and it is completely separated from the cytosol, surrounding cytosol, by the membrane. The composition of this luminal space inside the membrane is completely different from the cytosolic content of the cell. The endoplasmic reticulum is basically divided into two subcompartments. One is called as rough endoplasmic reticulum and another one is called as the smooth endoplasmic reticulum. Now, as we can see in the diagram, the rough endoplasmic reticulum is basically composed of the network of flattened sac. Okay, as we can see, there is a complex network of this flat sac, which are called as cristernae. Okay, and as you can see in the diagram, the endoplasmic reticulum is continuous with the outer membrane of the nuclear envelope. So you can see it is directly starting from the nuclear envelope only. On the other hand, if we see a smooth endoplasmic reticulum, they are highly curved. Here we have this smooth endoplasmic reticulum, which are highly curved and cubular in their structure, forming the interconnecting system of the pipelines traversing the cytoplasm. As we have seen in the previous slide, the diagram which were there in the previous slide, the rough endoplasmic reticulum has the presence of the ribosomes, which is bound to its cytosolic surface. Okay, the side which is facing towards the cytoplasm of the cell, or we can say the outer side of the cristernae. Whereas smooth endoplasmic reticulum does not carry or does not have the ribosomes bound on it. Okay. Apart from that, in the previous diagram, we have seen these rough endoplasmic reticulum. They are made up of flattened disc and flattened sac, and each of this sac is connected to each other. There is a continuous network of the sac, right? So this interconnection, or we can say these sacs are connected to each other by a membrane called as helicoidal membrane, okay, also known as helicoidal wrap, which we can see in the second diagram, okay. And as I have said in the previous slide, that smooth endoplasmic reticulum is highly curved. It forms a interconnected networks of the tubules. Okay, it is like a pipelines only. Okay, so that diagram also you can see over here on the on the slide. Okay, the first diagram represent the endoplasmic reticulum. Okay, now normally when we homogenize the cell by centrifugation, okay, so what happens, whatever membrane bound organelles are there, so the membrane gets separated, and when the membrane is separated from this membrane enclosed organelle, it gets linearized due to homogenization. But tendency of the membrane is to refold again, okay, and form a vesicle. So if I am homogenizing the cell, we all know the rough ER and the smooth ER, they have their own membrane, right? So when the cell is homogenized, the membranes of the smooth ER tubules will form a smooth surface vesicle because it does not have ribosome, right? On the other hand, the rough ER membranes will form a rough surface vesicle, which is due to the presence of ribosome. Uh, so when the study was carried out, the fluorescently labeled protein and lipids were tracked through the endoplasmic reticulum. And it is being observed that these labeled proteins are and lipids, they are capable of diffusing from one type of the endoplasmic reticulum to another. So it indicates that these channels, membranes are continuous and they are interconnected. Okay. In fact, these two types of endoplasmic reticulum, they share many of the same proteins. They are engaged in common type of the functions such as synthesis of lipid or cholesterol. And at the same time, you will see that in one type of ER, there are certain proteins which are present in larger number than the another type. Okay. For example, the high degree of curvature, we have seen smooth ER, right? So smooth ER tubules are highly curved, right? So this high degree of curvature of the smooth ER tubules is basically induced and maintained by a special type of proteins called as membrane bending protein, and which are basically nothing but reticulons. Okay, so this type of proteins are more in number in case of smooth endoplasmic reticulum. Whereas on rough endoplasmic reticulum, you will see they are hardly present in few numbers on the edge of the flattened sac. On the another side, the rough endoplasmic reticulum, it contains high level of protein which is involved in the movement of the nascent protein into the ER lumen, because it is mainly involved in the protein segregation and the protein uh, transportation. Then different types of the cell, if you see, they have different proportions of the types of the ER. Okay, depending upon the function which they are going to carry out. Okay, depending upon the function of the cell, the proportion of this ER will variable. For example, the cells which are involved in secretion of large number of proteins, like the pancreas, cells of the pancreas, or a salivary gland. So here obviously the proportion of the extensive region of the rough endoplasmic will be there. The proportion of RER will be more as compared to the smooth ER.